So maybe we can get rolling here in a second. So our topic for the next 2 or 3 of these is the immune system. And I hope, obviously, you know your immune system's role is to protect you from what we might call pathogens, germs. What are some things that can act as germs, pathogens for humans? Viruses, bacteria, keep going. Fungi, keep going. Some protists, like amoeba, certain kinds of amoeba, some types of dysentery, and there's that brain-eating amoeba that's been in the news lately. I can think of one other thing. Actually, I can think of a couple of things, but one other obvious. So they said protists. You're thinking of shows like Monsters Inside Me, what might you be thinking about? Such as worms. Okay, different kinds of worms like tapeworms and hookworms, and there's lots of worms. So different things that might be pathogens, bacteria and viruses, the 2 obvious ones. Fungi, maybe not as obvious. Protists, and there's all kinds of worms that can make you sick. Your immune system, its main job is to help to protect you from all of this. Now, there's 2 kinds of immunity that we're going to talk about. The non-specific, or sometimes it's called innate, and then also the specific immunities. What's the innate mean?
It's natural.
Yeah, it's like almost like an instinct. It's like built in, right? It's like hardwired, programmed. So this is this part of the immunity that we're going to start with is the most simplistic part of your immune system. It's called non-specific because it protects you against pretty much everything in general. No specific thing, just everything in general. And there's lots of components of your innate or non-specific immune system, but by far the most important part is your skin. Obviously, your skin's a barrier. It's like a fence, essentially. It keeps lots of things out, while it's also keeping what in? What's the most important thing your skin keeps it in? Your water. Keeps you from dehydrating. So that's one way, but it's more than just a barrier. It also makes all kinds of secretions. It makes some acidic excretions, things like sweat. And a lot of those secretions also create antibacterial chemicals in them to kill off germs. Sweat also has urea in it, which we talked about in here several times. What's urea? Somebody help me out. What's urea? Given off when you digest what? Proteins. And it used to be what? What was it originally in your body? What did your liver change into urea? Ammonia, right? It's pretty toxic stuff. So some of that stuff is put out into your skin. If it's toxic to you, it's also toxic to what? Bacteria and germs. So by having that stuff on your skin, you're sort of poisoning off some of the germs. You're killing them with your poisons. So that's part of your non-specific immune system. I'm going to skip through a couple of these. I want to jump down to inflammation next. When did you get inflammation? Cut. Any kind of trauma, right? A cut, a bruise, a bump. Things swell. Why do they swell? Why do they get inflamed? When you damage tissues, some of those tissues, especially connective tissue cells, send out like an SOS signal, damage signal. And that SOS signal you've heard of before. You may not know what it is, but you've heard of it. You've heard of histamine before, right? Where have you heard of histamines? You heard of antihistamines, which are medicines which help to cut down on what? Inflammation. Histamines, though, are essentially an SOS signal sent out by damaged cells. And what does that signal tell your body to do? It tells the blood vessels in that area to do that, which is what? What's that called? Dilation, right? Vasodilation. And at first that sounds like a bad idea, to send more blood to a place that's cut or bruised or damaged. But that's what you do, because what are you trying to send to those places? What do you need to get to places that are cut or bruised or damaged? You need platelets to cause clots. You also need white blood cells to kill whatever might have got in you when you damaged it, right? Whether it's bacteria or virus or protost or worm or whatever. So histamines, to a little bit lesser degree, you've got these similar chemicals called prostaglandins. And again, they cause the blood vessels in the area of damage to dilate. They send more blood to the damaged areas, to send more platelets to cause clotting, white blood cells to kill germs. Going down a little bit, let's talk about this one called interferon. There's lots of protein. The word looks a lot like interfere. Anybody have an idea what it might interfere with? A chemical made in your body that interferes with virus replication. Helps to mess up what cycle? The lytic cycle. So it's a way for your body to fight viruses. It's a natural chemical in your body that interferes with the lytic cycle, the virus reproduction. Viruses in general, not a specific kind of virus, but just viruses in general.
So, you know how you said separate viruses and free viruses, so we just get free doses.
They can give that. And they do to HIV patients and stuff like that.
So the difference is
Yeah, it's, I mean, with a normal, like just a normal stomach flu or something like that, it's probably not worth the money to do that. A lot of people wouldn't think of stomach acid as part of your immune system. But let's say when you're a kid, I have a 7-month-old baby. Think of all the things that go into a 7-month-old baby's mouth. Okay? Anything they can get a hold of goes into their mouth, right? Toys, bottles, whatever, the carpet, whatever they can get their mouth on goes into their mouth. And every time those things go into their mouth, germs go in. Luckily, where do those germs go first? The stomach, which is filled with what acid? What's the more science name for it though? Which acid is it? If we looked it up in the chemistry lab back here in the back, we would be looking at hydrochloric acid. And the pH in there is about what? 2 or so. Really acidic. What do we know really acidic things do to proteins? They cause them to denature. Well, what's a virus made out of mostly? Proteins. What's a fungus made out of? Proteins. What's a bacteria made out of? Proteins. What's a worm made out of? Proteins. So your digestive juices, your stomach acid in particular, kills a lot of the germs that come into your mouth. And that's a good thing. It's a really important part just of your non-specific immune system. A word came up the other day when we were talking about ecology in class, flora. And what did we say that meant? Flora. Normally it means the plant life in a place. Well, do you have plant life living in you? I hope not. But what might we call your normal flora? What do you have living in and on you? Bacteria. So when we talk about the body's normal flora, we're talking about the normal bacteria that live inside you. And they actually help to protect you. Let's take a normal healthy woman, for example. The reproductive tract of a normal woman is covered in bacteria. Let's say that woman takes antibiotics for something else, like strep throat. What a lot of times will happen after that? The woman will get like a yeast infection because you kill off the normal bacteria, that are just there and blocking other things out. Think of it like your offensive line. Those normal bacteria there blocking for you. And all of a sudden you kill your offensive line. Well, what happens to the quarterback in that case? He gets sacked, okay? And that's what happens when you're on antibiotics for something else. A lot of times you'll kill off the good bacteria that are helping you, and something that's not usually going to grow there starts to grow, like yeast. Same kind of thing can happen in your digestive system. What happens to a lot of people when they're on antibiotics? It's a side effect of antibiotics for a lot of people. They get diarrhea, sometimes vomiting. Because those antibiotics not only kill the germ that you're trying to kill, they also kill the good, normal flora in your intestines, which are blocking other things from growing there. And they help you not to get sick. Your saliva is also important. We can add tears here too. So let's say tears and saliva. Both of them have a protein in them called lysozyme. Tears and saliva. We're just looking at that. What does it sound like? It sounds like an enzyme that does what to things? Breaks things down. Hold that idea for just a minute. Hello? Hey. Yeah. All right, so back to what I was saying here. Lysozyme is this chemical that breaks things down. So it's in your saliva, it's in your tears. Think about how many times during a day you stick your finger up, you rub your eye, or you've got something in it, and you kind of poke it with your fingers. You probably didn't go wash your hands before you did that. Think about all the things that not being on your fingers. How many of you have ever had an eye infection? 2, 3, count me. And I'm going to bet I wear contacts. Either one of you guys wear contacts? Most of the people who get eye infections, like pink eye, wear contacts. You know what? Because you're constantly putting things in your eyes. Eye infections are really, really rare. Infections in your mouth, pretty rare. And the reason for that, one of the reasons, is because of this stuff. It helps to just kill germs, bacteria and viruses. Your mucous membranes. Where do you have those? Your nose, even your ears. Pretty much any opening into your body has mucous membranes, right? Urinary openings or conductive openings, nasal openings. What do those mucous membranes do? What's the main function of mucus? Catch stuff, right? It acts sort of like a filter. It's like a filter. Job is to trap things before they get too far into your body. And at least the respiratory mucus, where does it ultimately end up? In your stomach. So that those germs are killed right there by stomach acid pretty quickly. Going on down the list a little bit, let's talk about pyrogens. Pyrogens are chemicals that, what's pyro mean? Fire. These are chemicals that cause your body to raise its temperature, cause you to get a fever. A lot of people, as soon as they get a fever, what do? They go take Advil or Motrin or Tylenol or something like that. I'm going to tell you that by doing that, what are you really doing? You're making yourself be sick longer. Because the fever is actually not caused by the germ, it's caused by your body. It's part of your immune response. And when your fever's up a little bit, what works better? What works better when your fever's high? Your immune system works better. So it helps you kill the germ off faster. So by taking that Advil or that Tylenol, you might actually make the infection last longer. Now you'll feel better. Because when you have a fever, you feel cruddy, right? I mean, you feel tired and weak and chilled and all those kinds of things. But you might actually make yourself be sick longer by getting rid of the fever. The fever is actually a good thing, as long as it's not like 104 or something. All right, so those are just some of the parts of what we call your non-specific immune system. The parts of your immune system that just protect against things in general. But what we're going to talk about for the rest of our time today is what's called the specific, or sometimes it's called acquired immunity. If it's acquired, what does that mean? You have to get it, right? Who in here probably has the best immune system? Probably me. Because I've had more things. Been around longer. Fought more battles and won, right? And every time you fight a battle and win in terms of your immune system, what gets left behind? And you have these cells left behind called memory cells. And those memory cells remember what that particular germ looks like, and maybe more importantly, how to act, how to fight it. And if that germ ever shows up in me again, what happens? I kill it so fast that what never happens? That I don't ever get any symptoms. And so every time you get an infection, that happens. You build up all these memory cells. And that's why you don't get the same cold more than once. You don't get the same flu more than once. Now, you may get the flu more than one time in your life. More than likely you will. But what can we say about those different flu outbreaks you get? They're caused by different viruses. You're not going to get the same virus more than once unless something's wrong with your immune system. The first kind of immune response we're going to talk about, and actually before I get there, let's make sure we know these terms. What is an antigen? That's a word we used in here before a couple of times. It's a protein that's what? Foreign to your body. It's a protein that's not supposed to be there. It's not supposed to be in you. In response to having foreign antigens in your body, what does your body make to help you get rid of them? You make antibodies, your own proteins. So make sure you understand that we make antibodies in response to specific antigens. Now let's talk about the first kind of specific immune response, which is called antibody-mediated, or sometimes it's called humoral immunity. Two different names for the same thing. Back in like the old Greek times, they used to talk about the humors of the body. Anybody know what those were? The humors. Say again? You're in the right ballpark.
Your body fluids.
Yeah, like exactly. Your body's fluids, humors. Antibody-mediated immunity is called humoral.
Clinton Stewart, please go to Ms. McCutcheon's office. Clinton Stewart, please go to Ms. McCutcheon's office.
They call it humoral because antibodies are these big old proteins. When your body makes them, can they get inside your cells and fight infections that are already inside cells? What do we know about proteins and proteins getting into cells? They're not good at that, right? They're hard to do that. So antibody-mediated immunity, these antibodies can only kill germs if they're where? In the fluids, which would mean in the blood or in the fluids between your cells. That's the only time antibodies can help. They can help to kill germs that are either in the blood or in the fluids between the cells. They can't help with what kind of infections? What kind of infections get into your cells? Yeah, but what kind of germ usually does that? Virus. They can't help when the virus is already inside. They can get rid of viruses that are floating around out there, but if it's already in a cell, antibodies aren't going to help. And that's why we have another part of our immune system we'll get to either today or next time that deals with that. We've got two parts working together. This stuff we talked mostly about when we talked about cell signaling. This is another review of cell signaling. When a germ gets into your body, first of all, some kind of phagocytic cell. Usually it's a cell called a macrophage. And what do we know that word means, macrophage? Big eater. It'll come along and it'll bump into that germ that's in your body, a foreign antigen. And it'll phagocytize it, it'll pull it in. And when it pulls it in, what does it put it inside? What happens after something gets phagocytized? It gets put in a vesicle, right? And then what comes over to that vesicle and dumps in enzymes? Lysosome. And that breaks down that germ, kills it. And what do we call that process of dumping enzymes in there and killing that germ? Intracellular digestion, right? Now, most of the proteins that the germ was made out of, what's that cell probably going to do with them? It's probably going to reuse them, right? It's going to recycle them. But some of the proteins from that germ get put where? on the outside of that macrophage. We talked about, we call it almost like a flagpole or a trophy hand. It's called an MHC class 2 protein. So if this macrophage kills a germ, it takes a protein or a series of proteins from that germ and it essentially holds them up on the outside of the cell membrane like this. Saying, hey, this was in me. I killed it. I broke it down. But this is what it looks like. Who's it telling? What's it telling? This is what it looks like. Mostly it's the helper T cells. Those helper T cells will come in and they'll actually dock, physically dock with that MHC class 2 protein. And we said it was sort of like going Helen Keller on it, right? It goes in and it checks out that antigen, sees how it's shaped, figures out what it looks like. And then that actually activates that helper T cell. It's a form of local cell signaling. And they actually have to physically touch each other. The MHC class 2 and the helper T cell, they dock together. And once they do that and the macrophage gets activated, it starts sending out a series of its own chemicals, signals. And those signals are usually called interleukins. What would that mean, interleukin? Let's break that into pieces. What's that word literally mean? Interleukin. Between, what's leuco or leukin mean? What's a leukocyte? Different. It means white. So this is like a between white blood cells. It's a signal between white blood cells. So essentially, docking with this MHC class 2 activates that helper T. And then it sends out all kinds of signals. What historical figure did I compare a helper T cell to? Paul Revere. Sending out the signal. It's not so much the germs are coming, it's what? The germs are already here, and here's what they look like. That's what this cell is sending out. The germs are already here. Here's what they look like. Kill them. That's essentially what the helper T cell is doing. Now, some of those signals, those interleukins, will go out and turn on other helper T cells. And that's important. But another thing that happens, a lot of these interleukins will go out and activate a group of white blood cells called B cells. And those are the ones that are crucial for what's going on here. They help to turn on, after they dock with them, a set of B cells with matching receptors. Receptors that match what? What do these receptors have to match? An antigen, right? The foreign protein, the piece of the germ. And the B cells with those matching receptors, a lot of those B cells, I'm going to go to the next slide, basically get turned on. B cells or B lymphocytes with receptors specific for that antigen, most of them turn into what are called plasma cells. They swell up, these B cells do, and turn into these cells called plasma cells. And they start making thousands and thousands of antibodies every minute. Antibodies that are specific to what? That specific piece of that specific germ, that specific antigen. Now also, some of the activated B cells, though, don't ever become plasma cells. What do some of the activated B cells become? Can help you later. Some of them become memory cells. They remember what that germ looks like, and they can kill it the next time if you get it again. We'll talk more about this middle phrase in a minute. Some of you got the information you need to know to understand this today. When scientists first started working on the Human Genome Project, how many different genes did they think they would find in humans? Thousands and thousands. But what did they actually find? There aren't so many. And it turns out there are very few genes in our body that allow us to make antibodies. But if you were in class today, at least in 2A, what did we find out about one gene? it can actually, depending on how it's processed, it can code for multiple what? Multiple proteins. One gene can actually come from more than one kind of protein, depending on how it's spliced. And that's what all this is talking about. We'll get more details of that a little bit later on after everybody understands the genetics behind it. Let's talk a little bit about antibodies as we go here. Do antibodies actually kill a germ? Not usually. Antibodies can do a couple things. One thing they can do is let's say that action over here is a germ. Imagine that, right? Action is a germ. And this is an antibody specific to action. So it sticks to action. And think of this like a beacon. Think of it like a flashing light. And it's saying germ, 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 danger, 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 danger. And who's it telling that to? Yeah, what are the kind of the big dumb killers of your immune system are those macrophages. And most of the time they're just kind of floating around randomly bumping into things and killing them if they need to. But if there's a big flashy light on it, what do? They go to it and they kill it. It helps them kill it fast. So that's one thing. It can actually be sort of like a mark for death. So that's one thing an antibody can do. It can be like a mark. Another thing is it can be sort of like, what does the doctor tell you if you have like a virus infection? What does he tell you to do? Go home, get some sleep, and do what? Drink plenty of fluids. Well, in this case, let's say Lindsay is a germ. Again, imagine that, right? So the antibody sticks to Lindsay. There are another way an antibody can work is it can act sort of like a handle. If she's dangerous and she's a virus, and I'm a white blood cell, then I want to touch her. Yeah, right? So once this antibody binds to her, it can be sort of like a handle. And as another kind of white blood cell, I can grab that and drag her around. And where am I dragging her to? I'm dragging her to the kidneys, which filter her out, and then you just flush her out in the urine, right? And that's why you need to drink a lot of fluids. Flush out a virus. They get the antibodies acting sort of like a handle, and you're essentially dragging it to the urinary system. Another thing antibodies can cause germs to do is it can cause them to agglutinate. What's that mean, agglutinate? It means like clump up. So you knot them up, and that makes them easy to kill. So essentially, what antibodies can do is they can be like a mark for death. They can signal other parts of your immune system to that antigen so they quickly kill it. It can act sort of like a handle, so the parts of the white blood cell can basically escort that germ to the urinary system and filter it out. Or it can help to like clot up. And yeah, adhese might be a good word, so that it kills them. And remember that after the pathogens are gone, after the germs are killed, you've got those memory B cells that are left behind. So that if you ever get that specific germ again, your body will very quickly react to it and kill it before you ever even get any symptoms. I want to go to a little flow chart here first. Actually, before I do, this is supposed to be an MHC class 2 protein. What kinds of cells in your body have MHC class 2 proteins? Only the cells that are like phagocytic, that eat germs and then do what with the pieces? Stick them out there. What's that called? When they take an antigen from a germ and then stick it out there. Antigen presentation, right? That's a type of cell signal. They stick it out there, and then what do we say has to dock to it and go helter-skelter on it? The helper T's. And that helps to activate them, and those helper T's in turn send out their own signals, which activate other parts of your immune system. Let's go to a picture over here. Actually, before we get to the picture, this is what an antibody looks like. An antibody is this big old protein. And I want you to notice the most important part of the antibody to pay attention to is this part right here. What does that sort of look like? That we've talked about over and over again in here. This little groove. Think about it, enzymes. What does it sort of look like? So it looks like an active site. What do we know about active sites and substrates? They have to fit like a what? Like a lock and key. Well, this antibody's made so that that groove will only fit on a very specific way. What do we say? Antibody has to fit to antigen. So an antibody against this year's flu virus, is it likely to help you against next year's flu virus? Probably not. Now, if this year's is still going around next year, then yeah, you're probably going to be okay. But it's probably not going to help you if the flu virus has changed very much, because they're very specific. That's why this is the specific immune system. Let me get to a diagram here real quick. All right, this is trying to basically show you the process that we just talked about, the second picture here. What would the purple thing here be? That's the phagocytic cell. That's your macrophage. Notice it's eating the germ. The germ sort of looks eating soy, I think. But it's eating the germ. And then what do we say it does to that germ after it eats it? Puts it in a vesicle, turns what loose on it? Enzymes from the lysosome. What's the process? Intracellular digestion. And then it takes what kinds of pieces from those germs? Takes certain kinds of proteins, certain kinds of, what's the other word? Antigens. And it puts them out here on this green thing. What's this green thing? MHC class 2. And the process of taking those proteins and putting them out there on that MHC class 2 is called what? Antigen presentation. And who is it presenting the antigens to, or what is it presenting it to? Helper T. So this thing right here would be a helper T. And notice it comes in and it binds to, it physically connects with that MHC class 2. Like I said, it sort of goes Helen Keller on it. It figures out what the antigen is shaped like, what it looks like. And then it starts sending out its own signals, which are called what do we say those signals, the helper T cells send out? Interleukins. And those activate other helper T cells. More importantly, they activate cells called B cells. And we'll talk about that on the next slide. This is a really good diagram right here. And it sort of talks about both parts of your specific immune system. We're not going to get to the second part today. The left-hand side is the part we've been talking about so far. Humoral or antibody-mediated immunity. First of all, you've got an antigen in your body, some kind of germ. It gets engulfed by a macrophage. The macrophage destroys it and displays its antigens. The helper T cell docks to that, and it gets turned on. That helper T cell then sends out interleukins, cell signals, and those stimulate B cells. A lot of those stimulated B cells become plasma cells, and they start chugging out thousands and thousands and thousands of what? What do we need a plasma cell to make? Read the slide. Antibodies. Antibodies specific for that what? For that antigen. And they'll help you to kill it, help you to get rid of it. Also remember that some of those B cells don't become plasma cells at all. Some of them stay behind as memory cells. So that if you ever happen to get that infection again, you've got a plan. You've got some defense. Now, we're not going to have time to get too detailed with the other kind of immunity today, but I'll give you an overview and then we'll call it quits. The other part of your specific immune system is called cell-mediated immunity. We said that the antibody-mediated immunity, those antibodies can only get to germs if they're where? In the fluids, between the cells, in the blood. But infections like viruses, where do they spend most of their time? Inside cells. Antibodies can't get there. You need a way to get rid of infections that are internal, intracellular infections. And that's where cell-mediated immunity comes in. Its job is to actually kill what? Your own cells that are what? Sick, so that they can't what? Yeah, so they can't spread. Because typically, what's the most common intracellular infection? Viruses. And what do we know those viruses are doing while they're inside your cells? They are reproducing through what process? Lytic cycle. And given enough time, what's going to happen to that cell? It's going to burst, and out are going to come what? Thousands more viruses. So you better do what if you want to stop the infection? You better kill that cell before that virus has enough time to reproduce, right? That's what cell-mediated immunity does. And I'll mention this cell signaling bit, and then like I said, we'll call it a day. How does your immune system know that a cell's sick? Every cell has on its surface a protein called an MHC class 1 protein. And normally, what's it doing with that MHC class 1? Every day, your cells are taking some of the proteins from inside themselves and presenting them outside on that MHC class 1. Everybody takes something and hold it up in your hand. Doesn't matter what it is. So, parts of your immune system called cytotoxic T cells and also the helper T cells are coming around and checking out these MHC class 1s. And most of you look like you got the same kind of protein sticking out, right? What are these pens and pencils probably? If we're talking about color cells, what would these pens and pencils be? Just normal old proteins, right? Normal old human proteins in this case. But then we come over here, and we got a couple of oddballs over here. Okay, here's an oddball. There's a really oddball. There's some more oddballs over there. So if I'm a helper T cell or a cytotoxic T cell, and I attach to this thing, that's not normal. And I attach to this thing, that's really not normal. What does that signal me to do? That either signals me to kill that cell or to signal something else to come kill it. Because that tells me there's some proteins coming from within that cell that aren't supposed to be in that cell. And what are those proteins likely to be if they're coming from in a cell and they're not supposed to be in that cell? They're probably virus proteins. So that's the job of cell-mediated immunity. Its job is to kill your own cells that are sick so they can't spread that infection. Does that make sense? That's a good place to stop for today.